Thank you for joining us today, and thank you to all IAE members who support our organization. If you are not already a member, we invite you to visit our website and join the IIA. But our special thanks today go to Dr. John Paul Zonneveld, who gently agreed to share with us his insights about trace fossils in bones and their paleoecological value. John Paul Zonneveld, who we friendly call JP, is a professor in the Department of Earth and Atmospheric Science at the University of Alberta and curator of the University of Alberta Core Collection. After receiving his PhD from the University of Alberta, JP spent 2 years as a postdoc at the University of Calgary and then worked at the Geological Survey of Canada from 2000 to 2008, when he accepted his present posting at the University of Alberta. JP is a field-based research focused in the areas of sedimentology, stratigraphy, paleoecology, and paleontology. John Paul's research projects are typically multidisciplinary and commonly focus on problems involving the interface between geological and biological systems. His current projects include the sedimentology, biostratigraphy, and paleoecology of Mesocentric deposits worldwide, and the vertebrate paleontology of Paleogene and Quaternary successions in Wyoming and Maritime Southeast Asia. This talk will be recorded, thus I gently ask you to close your microphones to prevent noise interference. JP, you have the floor.
Okay. Thank you very much. Thanks very much for the invitation today. It's great to talk to you folks. I want to talk about the Ichnology of Living Bone. It's kind of a topic that's very near and dear to my heart. I've been a ichnologist and paleontologist for 30-something years now. And when I first started my career, I was working on fossil vertebrates. That was my original passion. When I started working with Dr. Alan Holman at Michigan State University, Alan would take me out to different localities to help collect herps for the Michigan State collections. And so we'd go out in the summer and fall and collect turtles, lizards, snakes, you name it. And one of the things I noted at the time, long before I actually had ever even heard of the discipline of ichnology, but one of the things I noted was that many of these turtles actually had parasites on them, most commonly leeches. One of my jobs was to actually take the turtles, especially after winter kill resulted in a whole bunch of mass mortality events in Michigan. It was a common event. We would collect all of the dead turtles, and my job was to actually turn those turtles into skeletons. And many of the turtles I noted had anomalies on their shells, pits and holes and scrapes and you name it. And I'd made notes on that, took copies of photographs of the different traces, and retain that. And flash forward a couple of decades, and I had been introduced to the discipline of ichnology and started to think, well, I remember all those turtles I saw back then. What's going on with that? What happened with that? And when I went through my photographs, I noted with a lot of living turtles, I could actually see parasites in those photographs. I could see that different things had attached. And so that's what got me interested and start working on bone as a substrate. Oh, and I'm realizing I don't have the controls to move my slides. Oh, so here's the next slide here, but I'm not sure who advanced that. Anyway, historically, we've actually accepted 3 substrates with taxonomic significance: sediment, rock, which includes shell, and wood. And I'm not able to move my slides. Slide control, what's going on here?
We see the second slide.
Okay, yeah, we can see it, but it's not advancing for some reason.
Okay.
So recently, workers have argued that bone comprises a unique substrate. It's distinct from invertebrate shells and rock substrates. And I'm one of those researchers that believes that bone really is different. I'd argue that bone is not the same thing as a shell or a rock. It actually exhibits unique challenges to animals that bore in. And this is especially true when we start thinking that there are certain types of bone that can actually be accessed when the animal's still living. And that, of course, is osteoderm material, dominantly osteoderm material. And in the cases of some animals, skull material. Sea turtles, for example, have a very, very thin layer of epidermal scute over top of their skulls. So a thin layer of epidermis underlain by a thin layer of collagen, and then the skull. And so the bone is actually easily accessible by parasites during the lifespan of the animal. And I'm still not able to advance. Are you able to advance for me?
No. Maybe at the bottom left, when you moved your cursor, there was something at the bottom left.
Oh, there we go.
Yeah.
There we go. Okay, that's perfect. Wonderful. So what is bone made of? Well, bone's made of dominantly hydroxyapatite. It's a calcium phosphate mineral. It's a lot different than calcium carbonate, which is what the dominant composition of most invertebrate shells will be made of. It also consists of 10 to 20% water, and living bone actually contains a huge amount of collagen, other proteins, and inorganic salts. Now, these components are not distributed evenly, so most bone has a denser outer cortical layer, a inner more cancellous spongy zone, and then, of course, whatever organics are present in there as well. During lifetime, there would, of course, be a layer over top of this, and that would be that epidermal scute layer that I was referring to, as well as a collagen layer. Any animal that wants to penetrate living bone has to actually get through those types of substrates. Another important thing to consider is what is a pathology and what is a trace, especially when we're dealing with things that actually access living bone. I'm pretty simple with this, and this is actually an important issue for me because every time I submit a paper to a journal, I have a veterinarian type or a biologist type researcher that argues with me, well, why are you not treating these as pathologies versus trace fossils? Well, I would argue that pathologies result from the host response to a stimulus. And so bone spurs due to osteoarthritis, galls on crinoids, things along those lines. It's what the animal, the host animal, is doing and not what the external agent's doing. In contrast, trace fossils would derive solely from the actions of external agents, beetle borings, snail borings, etc. The problem we have with taxonomy in traces that are in place in living bone is that the actual trace that's being emplaced by a leech or a tick or a flea often prompts a systemic response from the host. And so most of the time when we're looking at living bone, we're not looking at things like the top right, which is a cervid mandible that was gnawed on by a porcupine. We're not dealing solely with things like that on the left, which is actually a cancerous osteoblastoma in a turtle shell. We're dealing with something that's actually a hybrid, something in between. And so we can name, of course, the trace fossil, but we may not name the pathology according to the ICZN rules. As I'd mentioned, bone presents challenges to trace makers. The trace maker has to be able to attach to a living host long enough to penetrate and affect the bone. And they have to be able to avoid removal by the environment or by the host. Many hosts can actually groom themselves. The trace makers have to be uniquely adapted to penetrate through very, very distinctive substrates. And as I mentioned before, that includes the keratinous scute, the layer of collagen, and then into the dense cortical carapace and plastron bone of a turtle or the shell bone of a glyptodon or an armadillo. Another problem, of course, is that many hosts are amphibious. So the trace makers have to actually be able to either leave their mark very, very quickly on the bone or be able to survive both aquatic inundation and subaerial exposure. So there's a lot of challenges to penetrating into living bone. Now, what animals actually have osteoderms that can be penetrated? Well, extant organisms include chelonians, armadillos, crocodilians, lizards. I didn't include it here, but there's even some species of mammals outside of armadillos that actually have osteoderms, including some mice. Extinct animals include a lot more characters. Giant armadillos, so glyptodons, Boreostoma, Holmesina, etc. Ankylosaurs, temnospondyls, stegosaurs, phytosaurs, aidosaurs, placodonts, Hufasuchians. There's a lot of different characters that can actually have osteoderms that are potentially accessible to parasites. To date, the only ones we've actually seen evidence of parasites and fungal infections and bacterial infections, turtles, armadillos, and giant armadillos. Those are the only things we've actually found traces in so far. So I'll start off with talking about turtles and tortoises. And when we talk about turtles and tortoises, the most common things that are actually affecting the bone are things like leeches in aquatic animals, and then ticks and fleas have that potential in terrestrial organisms. Most of the time, we see hemispherical and columnar pits that can be pervasive on turtles, carapaces, and plastron. We see these both in terrestrial forms, so animals that live on either in water or on land in terrestrial settings. We also see these in sea turtle forms as well. We'll talk about both. Most common thing in aquatic terrestrial turtles are leeches. And leeches will attach to the epidermis using suckers, as we see on these two different turtles right in here. Both of these are North American forms. Top left is Trachemes. I believe the bottom right is actually another species of Trachemes, but it's more of a form with a darker shell. The leeches will attach using both their anterior and posterior suckers. And so we can see in top right there, two leeches attached to the plastron or bottom shell of a turtle. They can stay on for extensive periods of time. Their problem, of course, when they're on the plastron, that bottom shell, is being rubbed off on a rock or on dirt when the animal comes out to bask. In the bottom image is what we're seeing there are the proboscis of a leech, of a turtle leech, a Placabodella. And what we can see on there are a whole series of small pores labeled by P. And those pores right there are what actually emits the osteolytic enzymes that penetrate into the bone. And so we actually have to have a leech do two different things. One is mechanically penetrate through the epidermal scute. And then, subsequent to this, we actually emit the osteolytic enzymes into the penetration, which then corrodes into the bone. Now, after this, they may actually prompt a healing response by the turtle, and so we may actually see a change in bone density adjacent to the place where the leech is attached. In this case right here, we're looking at the bottom side of a Graptomys, juvenile Graptomys. In this case right here, the leech is actually attached to the joint in between two different epidermal scutes. We refer to those joints, those connection places, as sulci. And so in many cases, leech attachment traces will actually go along the sulci. Now, when we actually look at turtle shells, we look at the sulci rarely occur at the same joining location as the bone joints. And so we can actually see in the very center part of individual bone elements, we'll see individual lines of connection traces. Now, in some cases, that doesn't occur. In some cases, the leech is actually penetrating through the very center part of a scute. A question we have, and we still don't have an answer to, is this a tracemaker-specific behavior? Do we see randomly distributed traces belong to one tracemaker group and non-random ones, traces that actually occur along the sulci, to another tracemaker group? We don't know. We don't know if Placabodella can actually penetrate through the scute, or it's restricted to the epidermal sulci. This is a question we're still trying to answer. One of our problems, of course, is that we rarely actually, in fossilized material, see the scute material preserved. In modern material, it's rare for us to actually be able to look at both the location of a leech attachment as well as look at what happens to the underlying bone, because of course we'd have to kill the host animal in order to assess the trace. And so this is an area that we have a lot of work to do. Another example of animals that actually attach to turtles and tortoises are ticks. Now, ticks, of course, are terrestrial. They are air breathers, and they're not going to survive aquatic inundation very long. And so they're unlikely to be trace makers in aquatic turtles, but they certainly have been known to produce traces in modern terrestrial turtles, animals that rarely enter the water. And we've seen them on tortoises as well. And so what we're looking at right here, a pair of exotic ticks attached to the base of a terrestrial land-based turtle. These are a number of images, and I apologize for the lack of a scale on these images right here. Um, as I mentioned to the hosts of our presentation today, I had a bit of a technological glitch right before my talk. And so I spent the last hour replacing images back in my presentation. And so for any grad students out there, you should know that even for us seasoned professionals, glitches happen and last minute stuff is um, part of our daily existence. But anyway, when we're looking at this slide right in here, we can see in here that the anterior part of the plastron of a turtle. And so the top left image shows the bottom side, the external surface of the turtle. And we can see a heart-shaped image in there. And I'll see if I can actually annotate right in here. We can see this heart-shaped, where's my pen right there? And we can see an area in here. I'm hoping you guys can see my pen right in there. So you can see this heart-shaped chunk right in here, which is the anterior part of what's referred to as the epiplastron. We can also see in this area I'm circling right in here, a pair of small divots. And those divots right there are traces emplaced by some type of trace maker. And in this case, we actually believe they're actually ticks. We think, again, the same thing happened, that osteolytic enzymes are penetrating into the bone and allowing the tick to attach. It's essential on that bottom side for that tick to actually go into the bone because, of course, this area would actually rub against the ground as the tortoise is moving forward. The bottom left shows a close-up of those two traces. The images on the right-hand side show two chunks of bone, in this case here, on the interior side. And so this area in the bone above the line I'm drawing right now, this area would have actually been, sorry, I can't draw all the way, there we go. That area would have actually been exposed to the air during the lifespan of the animal, and the area below that would have been actually attached to integument. And again, right here on that lip, we're seeing two traces. We can show the close-up in the bottom right, again, of pits that are actually emplaced by some parasite, which we believe in this case here to be ticks. Okay, now that I've done that, I'm gonna have to go back. There we go. There we go. Okay. Moving on then, other things that can actually affect bone or include things like bacteria and fungi and algae. All three can corrode bone in different ways. They can be random or systematic. There's a couple of diseases that turtles get. There's a whole series of these. I'll refer to three right now. SCUD, subcutaneous ulcer disease, USD, ulcerative shell disease. Those all start off as tiny patches, but they can produce pervasive amorphous bone degradation on both plastron and carapace bone. And on the right-hand side, I'm showing some examples of different levels of ulcerative shell disease and subcutaneous ulcer disease. In the upper left images right there, we're seeing some areas of corrosion, amorphous zones of corrosion. In the other images, bottom left, we're seeing a turtle plastron, the complete turtle plastron, with huge zones where my scale is, as well as in the anterior part of the turtle, where we're actually seeing absolutely huge areas that have actually resulted in penetration by the etiological agent right through the bone. Now, that's an interesting problem right there. When the animal's shell is penetrated, could the animal survive this? They can, to an extent, but eventually it will actually weaken the bone to the point in which it will actually kill the turtle. So what causes that? Well, in this case right here, it's actually a mix of all three. Bacteria, fungus, and algae are actually involved in that. The interesting thing with that is that those agents are invariably present within turtles' environments. They're always present. And so it actually takes something else to cause this to result in significant bone corrosion. Something has to happen to the turtle's environment or to the turtle's health, some extra stressor to allow the turtle, to allow the parasite to penetrate through the turtle shell. And usually that's environmental degradation, increased stress in the organism itself. Any of these things can actually result in bone degradation. And so usually when we see this, something unusual has happened. Another example of a disease is called carapacial shell disease. And that's actually something produced by algae. And it produces linear to curved, very, very narrow tracks in the surface of the carapace. Moving on. There we go. This is an example of some fragments of turtle shell from the Eocene of Wyoming, and where we can actually see some pretty significant ulcerative shell disease. You'll note here that we can actually see healthy bone in gray I'll put my pen on again so we can see this line right in here separating healthy bone to the left, where we can actually see a epidermal sulcus. I'm drawing right here. That's an epidermal sulcus right there. That epidermal sulcus continues on into an area that has very, very ugly bone. We're going to actually see that sulcus. I'm drawing it on the bone right there. That sulcus continues on into the area that's actually been affected by significant bone degradation. And so in this case right here, it's ulcerative shell disease that didn't kill this particular turtle, but certainly had a dramatic effect on the shell of this turtle. Here's a close-up right in here. We can see healthy bone towards the right-hand side. And then on the left-hand side, again, I'm circling this out with my green pen. We can see corroded bone on that side right in there. And that bone corrosion can actually result in corrosion just on the very surface of the outer cortical layer, or it can penetrate from the outer cortical layer into underlying layers, and in severe cases can actually move on and completely penetrate through into the internal integument of the turtle. We've named some of these types of traces. Gunnolochnus mogrensis and Gunnolocus aculathistae are very shallow traces that tend to affect the outer surface of the cortical bone. In both cases, we believe they're bacterial or fungal trace makers. There's an example right here shown in the bottom right, both in the a whitened turtle shell with very, very low angle light because it penetrates only a fraction of a millimeter into the shell itself. We can see two patches on here, and these are referred to Gunnolochnus mogrensis. The second ichnotaxon I have listed here tends to actually have very, very narrow branching penetrations. Gunnolochnus mogrensis tends to actually be much more amorphous and pervasive. We could probably extend the use of this ichnotaxon into fossil cases where we have USD or SCUD, where we've actually got deeper penetration into the shells. More recently, we found a third type of gunnolochnus that actually occurs in long, narrow tracts. And we think these are actually not produced by bacteria or fungus. They only occur on the carapace, which is the upper shell of the turtle. They never occur on the plastron, the lower shell of the turtle. They're only occurring in areas that are accessible to significant light penetration. They're at the top surface of the turtle. So we actually think they're actually formed by different types of algae. To date, we've found around 20 specimens that actually show this material in multiple horizons. Again, it's from the Eocene of Wyoming, but we're actually seeing them in different depositional basins. We see them in the Green River Basin as well as the Wind River Basin. We see them on two taxa, Baptomys wyomingensis and Baptomys garmani. We have numerous other turtles, and we never see them on other turtles. And again, they only ever occur on the upper surface of the turtle, on the carapace of the turtle. This particular taxon differs from previously described species of gunnolochnus in the occurrence of all of these, and I'll draw them on again, all these hemispherical pits that we see on these turtles. So we can see one right here on that bottom image, all these individual pits on there that occur in association with that, as well as on the this piece of carapace on the left-hand side of the photograph. And again, numerous hemispherical pits occurring within in that. Like I said, we don't think these are actually related to fungus or bacteria. In this case right here, we actually think they are related to algae. And here's an example of a modern turtle, a modern pond turtle from Texas, where we can see these linear trends showing individual hemispherical pits of algae attaching to the outside of the turtle. In severe cases, this penetrates through the epidermis and affects the underlying bone, producing traces very, very similar to the traces I showed you from the Eocene of Wyoming just a few minutes ago. So moving on, not all hemispherical pits are the result of algae or ticks or leeches. We frequently will see hemispherical pits in fossil sea turtle and modern sea turtle. And so here are some examples of the sea turtle Caretta caretta. And we can see on this sea turtle numerous large and small barnacles. And barnacles can produce these pits as well. And so in the upper left image, we see some huge barnacles just behind the sea turtle's head in the very top part of the carapace. Those are a barnacle called Cheilonibia. Cheilonibia does not affect the underlying shell. It actually glues itself to the external surface of the epidermal layer and can move along. But if we actually look at the images in bottom left and top right, we're actually looking at zoomed in images along the carapace. we're actually looking at different traces, a different barnacle, sorry. And these barnacles are something called Platylepis and Stephanolipus. And these barnacles actually don't move. They actually penetrate into the shell of the turtle. And so they'll actually penetrate through the epidermis into the dermal layer, into the dermal bone. So top right image, we can see a whole series of these barnacles almost solely occurring in or adjacent to the epidermal sulci. And so that's that seam in between the keratinous epidermis. I spent some time working in the Smithsonian, and there's an amazing collection collected from Cumberland Island in Georgia that was collected by a woman named Carol Ruckstell. And she actually had the foresight not just to collect the bone of the turtles, the skeletons of the turtles, but she also collected the epidermal scutes as well, which was a wonderful thing. And so here's an example of the posterior half of a Caretta caretta, a large sea turtle, a loggerhead sea turtle. But you can see at the bottom right on the in that tray, we actually have one epidermal scute laying there. And the joy with that particular epidermal scute is that epidermal scute overlaid the bottom part, and I'll circle it right here. That epidermal scute would have actually occurred on this layer, this area right in here of the dermal bone right in there. And so that particular scute right there occurred in that area. And so we can see, I'm circling it right here, one large trace right there, a second large trace right here, both hemispherical pits. And so here we can see those two individual scutes. We can see right in here on this trace, and I'll go, sorry, backwards again. We can see on here, got my pen drawn here, we can see a couple of large traces right in there, drawn on there in green. Those traces were an area where a Cheilonibia tustudinaria would have attached. You'll notice in the same area on the bone, there are no traces. We can also see embedded within the epidermal layer, there and there, a couple of embedded barnacles. Those are both Platylepis. And we can see here and here where those Platylepis actually penetrated into the bone. Here's a plastron of the same individual. And again, we can see numerous areas in which we've actually got significant barnacle incrustation traces on the on this bone. And again, when we actually look at the epidermis, we can actually see the barnacles and identify the taxa that created those traces. We can also see on this particular sea turtle, there's an awful lot more traces on its plastron than were present on its carapace. At the right-hand side, I see I show two different traces. I'm not sure why I put them where I did, because this particular trace is this particular trace, and this particular trace is this particular trace. But what I wanted to point out is we have two different taxa of barnacles that actually had attached. We identified the two different taxa. Upper right-hand side, we have Stephanolipus. Bottom side, we have Platylepis. And in the bottom side, we can actually see that the trace drawn here in a large circle, I'll let that fade away, actually has a whole series of subpits in there. And that's unique to Platylepis because they actually have elements in their shells referred to as pillars that actually produce these 6 individual pits within the bottom part of the trace. So we can actually start to connect individual trace morphology with specific trace makers on these modern sea turtles. Here's a skull of Caretta caretta. And again, what we're looking at right here is the actual skull at top left. And we actually have the skull with the atomium in place, the keratinous beak that occurs on top of the sea turtle on the right-hand side. And in this case right here, we can see, and I'll draw again, individual barnacles. And these, in this case right here, they're all Platylepis on this particular turtle right here. And we can actually see the places on the skull where those traces actually emplaced pits, hemispherical pits, into the underlying bone. We can also see in here too, I'm going to just draw on the main skull right here, areas on the skull where we've actually got abraded pits into the skull bone where we actually had barnacles attached as well. Another example in here, and again, in this particular case, we had one single barnacle attached, embedded into the tomium of this turtle, and we can see that barnacle right here, and we can see an underlying pit. Now you'll note that it doesn't actually penetrate that deeply into the bone. In this case, it's because that tomium keratin is much thicker than an epidermal scute would be. So at left-hand side, we can see a caretta, a loggerhead sea turtle skull with numerous abrasion pits, numerous scars where bone scars where individual barnacles would have attached. Some are very, very shallow, like this one drawn right here. And some are much, much deeper, like this one right here. And you'll note that this particular one is actually at the suture line in the individual bones. On the right-hand side, we have a sketch of caretta skull, where we've actually drawn on the barnacles that were attached to 24 different sea turtles. And so we've actually been able to identify areas that are actually more likely to have barnacles placed on them, in areas in which they're less likely to be placed. And so you'll note that most of them are actually on the anterior half of the skull. They're in an area with significant water flow, which actually was going to produce more food. And you'll also note that a significant amount occur on the skull, but near the mandible itself. And so penetrating through the tomium into the skull bone in an area in which the trace maker was more likely to encounter food, right adjacent to the turtle's mouth. So moving on, we don't just find these hemispherical pits in sea turtles. We don't just find them in land turtles and sea turtles. We also find these in other shelled organisms. And here's a couple of examples of armadillos. Recent work by a professor, Maria Esquiaga, out of South America, has identified traces made by fleas on the carapace of different living armadillos. And these are the two that she actually worked with initially, and she's identified them through a whole series of other armadillos more recently. Here's a number of images from her paper where we can see a number of these pits on the the posterior portion of a carapace of Toleptes on the left-hand side. We can also see close-ups of individual traces on a number of different taxa on the right-hand side. More examples from her paper. Really, really beautiful work. And this is one of the very few papers written by modern biologists that actually show the bone traces themselves, a rare occurrence. Interestingly, we've actually found these in ancient armadillos as well. In this case right here, it's the giant armadillo, Boreostoma. What I'm showing right here are a couple of different osteoderm elements from a large Boreostoma. We see the tail at bottom right, and we see a single osteoderm on top right. Now, those holes in that osteoderm are actually follicles where hairs would have been, and those are not traces. But interestingly, we do find traces, and here's some examples right in here on Boreostoma osteoderms. Now, interestingly, in this particular example, we only ever find these traces, and I'm circling a few of them in here. They tend to occur again at the epidermal sulci, so in areas where the epidermis actually has a boundary between one epidermal scute and another one. And here we're actually seeing them at the conjoining areas in between individual bone elements. In Boreostoma, we only ever find these traces so far on the tail elements. And so they're actually on an area the animal would have actually had difficulty with grooming, and that probably would not have been abraded by passing through trees and brush and whatever else. And so the traces are actually able to attach and not become dislodged. Here's some traces on the caudal armor. These traces are generally very, very consistent in size. And so you'll note all the traces in this image, sorry, going backwards right here, all the traces in this image are roughly similar in size, very, very similar in their size, 2 to 3 millimeters in size. And that's consistent, going backwards here again, with the traces that were illustrated by Dr. Esquiaga on modern armadillos. So we believe these are also related to fleas. And in this case, the fleas are actually leaving traces that penetrate into the bone on the external surface of the carapace. So now we're looking at a image of these tail osteoderms as well. In this case right here, the caudal osteoderms. In this case right here, we're looking at some CT imagery, so we can actually take a look at the bone And this underscores that we're looking not just at traces, we're also looking at pathologies. And I'll point you to the arrows in here. I'm going to circle right there. Um, if we look closely at this particular trace right here, drawn right here, you'll notice a significant bone density difference as we go from the area away from the trace into the area where we actually have the trace itself that's blown up at bottom right. And so in this case right here, boring by the flea that made the traces resulted in a systemic response by the host organism's system itself. We've also seen this in Glyptodon. So here's some examples right here. Again, in this case right here, we're actually seeing a trace maker preference for the anterior and the lower portions of the carapace. We see fewer of these on the upper portion, more of these on the area right behind the skull, as well as on the lower portion. And that's probably because the wider portions and the higher portions were affected by the environment. When the animal passed through thick brush, those would be areas that would be more likely to dislodge the trace maker. Another example right here from Holmsina occidentalis. In this case right here, we actually see numerous traces, but in this case, we're actually seeing a wider variety of sizes, ranging in size from a millimeter. This particular osteoderm was about 2 centimeters across, and so the traces range in size from less than a millimeter across to more than 3 millimeters across. These might be flea-derived Corythricus lacos. They might actually be related to a different trace maker. And we have questions on this because we do know that ticks make traces in turtles. They've never been identified to make traces in armadillos, but the same taxa, at least the same genera that make pits within tortoise shell, have been known to colonize modern armadillos as well. And so we have a few questions then. Are all pits made in our modern armadillo initiated by ticks, or can fleas make them in armadillos as well? Another good question is, did the Tunga-armadillo relationship initiate in small armadillos and transfer to giant, or the other way around? And then at present, all of these traces are restricted to South America, even though giant armadillos and small armadillos make it into North America as well. And so are these traces strictly a South American phenomena, or did it cross the isthmus up into North America as well? Again, questions that we have no answers to at present. Other types of traces can be made by agonism. A recent paper, well, 10-year-old paper, 12-year-old paper, noted that in pachycephalosaurs, there are lesions. And those lesions occur in areas that are consistent from one animal to the next. And about 1/5 of these pachycephalosaur domes have these lesions. These lesions have been attributed to interspecific agonism, very, very similar to the types of scars that we see in the skulls of mountain goats and mountain sheep. This is a skull of a pachycephalosaur from the Hell Creek Formation that actually has a number of traces on it. We actually think this particular trace was the result of intraspecific agonism, resulting in a breach of the skin that was then colonized by a botfly. And the botfly larvae were actually making these pits into the top of the pachycephalosaur skull. So then the question arises, what about other animals with osteoderms? Do we see these in lizards? Do we see these in crocodilians? Well, the answer at present is not yet. I've spent an awful lot of time looking at crocodilian osteoderms. We have not yet seen any clear traces. Now, one of the problems we have with crocodilian osteoderm is they're a lot more morphologically complex than turtle shell or armadillo shell. There's a lot of pits, a lot of grooves, a lot of ridges on crocodilian osteoderm. But at present, we've seen nothing that is demonstrably a trace and placed by a parasite or a bacterium. We don't know why. We're not sure. I've looked at crocodilian osteoderm from hundreds of specimens, and I've yet to see a single thing that I was convinced was actually a trace. It might be that this is because of crocodilians' amazing ability to heal. They tend not to get bacterial infections. They tend not to actually get problems, even though they live in very organic, rich waters, even though they are animals that fight with each other. Almost all traces we find, all traces we find in crocodilians are clearly related to conspecific agonism or battles between different taxa. So why do we look at traces of turtles? Well, we're trying to actually understand more about their biology. We're trying to understand more about, sorry, when I say traces of turtles, why do we look at traces in osteoderm? We want to understand the biology better, but we also want to understand things about paleoecology better as well. This particular graph is from a paper we published recently where we were looking at ulcerative shell disease in turtles. We looked at ulcerative shell disease in turtles from a wide variety of localities in a large part of the Bridger Basin, which is a subcomponent of the Green River Basin in Wyoming. We looked at around 400 individual turtles to try and understand what was going on. And so what did we result in? Well, we found ulcerative shell disease in a few turtles from multiple horizons and a lot of turtles from one horizon. So we see right here an area where they're occurring on that single horizon in the middle of what we refer to as the RC bench. And that RC bench has a very, very thin horizon that produces abundant turtles. Interestingly, they occur dominantly in that horizon, and only a very, very few dominantly incipient cases, so mild cases of ulcerative shell disease in other horizons. All of the really severe cases, except for one turtle, occur in this one specific horizon. And this is again, this is after looking at hundreds of turtles. So the question to me then is, why is this? Well, one of the things we noted was we actually have significant volcanic ash in the layers in the middle RC bench, much more so than we actually see in other areas. And so our theory at the time was that we're actually looking at a time interval in which we had a lot more volcanism, resulting in probably more significant cloud cover, potentially more significant rainfall in the area, and turtle basking became less effective. And so the algae and the bacteria and the fungi that are natural in their system were able to actually affect the turtles in this horizon a lot more than other horizons. And that was a working theory that we had. That may or may not be true. Another thing that we saw, and this is actually now looking at data from two parts of the basin, the Bridger Basin and the Green River Basin proper, an area called South Pass on the western side. We've also noted that we've actually have a huge spike in ulcerative shell disease. It's actually occurring in the early Eocene climate optimum interval, an interval with greatly increased air temperature compared to other intervals. We also note that we actually see pits in turtle shell in South Pass greatly more abundant in the area in the same rough time interval during the heart of the EECO. And so then the question is, was this change in climate resulting in more susceptibility by turtles to parasites, where water's warmer, where trace is more abundant, where trace makers more abundant? We don't know the answers to this yet, but we are certainly seeing trends. There is an increase in susceptibility to shell disease and an increase in susceptibility to parasites within the EECO compared to horizons higher and lower. We are trying to expand our study in this. Right now, we've constrained our study to the Bridger Basin and to South Pass, where we've got great early Eocene deposits, but we want to expand this lower down to the earliest Eocene and higher up to the middle and upper Eocene. Are we seeing this trend? Can we actually tie this to climatic changes? It's a hypothesis. It's not been proven yet. And I mentioned before too, we're seeing regional trends in terms of the traces on armadillos. Right now, the only modern examples of these corythricness in modern armadillos are in Argentina and Paraguay. And that's the result of the excellent work by Dr. Escoge. Fossil examples are also restricted to South America. The oldest examples occur down in the Oligocene of Uruguay. There's also some examples from the Miocene of Argentina. And then more recently, we've had examples from the Miocene of Colombia. At present, they've not yet been described from anywhere outside of this area. Did they make it into Central America? Did they make it into Mexico? Did they make it into southern North America? We don't know yet. And so I'll leave with this. Looking at living bone, there are a number of challenges. And one of those challenges is the fact that we're actually looking at not just traces emplaced in a dead piece of shell, but we're actually looking at an interaction between a trace maker and its host. As a result, we're often looking at hybrid traces and pathologies. There's challenges in differentiating the traces that are in place between premortem, perimortem, postmortem bone. It's hard to tell that. A big challenge for us is that biologists and veterinarians very rarely consider the effect that those parasites have on bone. And as a consequence, illustrations of bone modification traces on modern bone are incredibly rare. Similarly, us ichnologists haven't done our job yet. We've done a few case studies, new ethnological studies of borers into living bone. The example of that would be the study that I did on sea turtles, looking at the effects of barnacles, and that was actually a great study because we were able to actually um, look at the embedded barnacles still remaining stuck within the epidermis and connect that to the bone. But we don't get that very often. To be frank, we've made inferences that may challenge the limit of our data sets. Our Trachemes paper, looking at modern Trachemes scripta, is a really interesting study, and we're inferring that leeches were the main trace makers. Now, we have actually seen leeches attached to some modern turtles that are actually associated with pitting. But the question then becomes, did those leeches actually enter those pits afterwards? Did they find themselves a place to shelter, or did they actually make those pits? And that's something that we will need to understand better. And so right now there are limitations to what we can say about the Ichnology of Living Bone. It certainly has great potential for saying some amazing things about paleoecological analysis, but we have a lot of work to do still to look at both modern and fossil bone to understand what are the distributions. At present, within ancient armadillos, we have a few case studies that look at one or two pieces of bone, a few pieces of bone, maybe a few hundred pieces of bone, but we need to do population scale analysis. We need to look at large scale collections of fossils to start to understand, are these traces limited to different areas? Do multiple trace makers colonize armadillo bone? More questions remain that have been answered so far. With that, I'll leave it for questions. Thank you very much for listening.
Thank you very much. It was a great talk. We have some time for questions, so you can open your microphones.
I see Lucas has his hand up. Oh, there we go.
Lucas.
Lucas, did you have a question? I don't see the chat. Let me see if we can see the chat there. Nothing in the chat. Okay.
No, there's nothing. No.
Okay. I saw Lucas, his hand came up for a second.
Okay, I have a question.
Hi.
Hi. Do you believe that we have too many ichnotaxa today? Because I see that difference between the ichnotaxa are not so there's not so much difference between them, and I see every day that a new paper with a new ichnotaxa. And so, you believe that we should reduce some of them?
Yeah, that's a great question. This is one of those things that Murray Ginger and I have talked about at length. I'm torn between whether or not we should be using Linnaean taxonomy for trace fossils because they're not animals, they're behaviors. One trace maker can make multiple types of traces. Um, and a single trace taxon can be made by multiple animals. So we have difficulty with that. Regardless of that, it is a system that we have, and it's a system that we stuck with. And so, in terms of whether or not we have too many ichnotaxa, the answer is yes and no. So I'll sit right in the fence there. I guess the question we have to ask ourselves whenever we erect a new taxon, is there a good reason for it? A good example is the Gunnolichnis that I was illustrating. When I first named Gunnolichnis on the Egyptian Miocene material, we had two very, very different forms, both very shallow penetration into the shell. And but one was amorphous, broad, often had scalloped edges. One was almost always linear and very, very narrow, and generally constrained to the area around the epidermal sulcus of aquatic turtles. They occurred both on plastron and carapace. And so that was two end member taxa. And I assumed when I named that, that those would be the only two taxa ever named. And then more recently, we found this other trace, what I refer to as type 3 in this talk. Um, and it's interesting because that particular trace only ever occurs on the carapace and almost only ever occurs in the carapace of one species of Baptomys with a couple specimens on a second species of Baptomys. In the same horizons, we find hundreds, more than that, we find tens of thousands of specimens of Ecmetemes. It never has that trace. And so now we've got a trace that occurs in linear trends, only occurs on the carapace of turtle, and in addition to being a shallow etching in the shell, also has hemispherical pits. So I could lump that into an existing species, but in this case, we actually have a trace that's probably made by a different trace maker. We think we've got pretty good evidence of that, and is taxon and site specific. And so I think there's actually benefit in naming a third trace because it's something distinct, it's something different, and it actually has different behavioral and ecological implications. So I guess my answer to your question is we should only be naming new taxa if there's a doggone good reason for it. It shows us something different, a different behavior. It shows us a different occurrence, something ecologically, paleontologically, biologically significant. You know,
Yeah.
I'll further with that. Corythricness, at some point, I think I'll probably revise that. I think I'll probably kill one of my ichnospecies in that because I think that actually there's a good evidence that that one should actually be simplified a little bit. Um, you know, one of our problems with ichnotaxa is that, you know, in one area, we may actually have a trace that has incredible distinctions. The morphology is absolutely distinct between two different end members with no overlap whatsoever in one population. An example of that would be corythricness. When I first named corythricness, we had hemispherical pits, we had cone-shaped and flask-shaped traces, and they were distinct. But when you look at other populations, different animals, and so the original corythricness were named in the turtle Ecmetemes. When we look at another taxon that we've been analyzing, now we actually see significant gradations between forms. Since I named corythricness originally, I think there's been two or three new species added to that. And again, I think we've overcomplicated things. And I don't think there's actually, I think we could probably reduce the current 5 or 6 ichnospecies down to 2 ichnospecies in that particular taxon.
Yeah, and I'm seeing also that some people are using Ichno family also that I don't agree, but the people are turning the Ichnology into a classic systematic, and I think that's not that's not fit because are different. As you said, Ichnology is about behavior. And a lot of people also do not use some parameters, such as the Ichnotaxobasis of Pironi. I believe that the classification, the identification of a lot of Ichnotaxa is not, it's not, does not have a standard. So I believe, that we should, how can I say, we should have a standard pattern for the identification and classification about morphology and the taxa. I don't know
I
if
think
you agree with me, but it's what I'm seeing.
I think you and I are actually in agreement on the taxonomy issues. You know, like I said, I'm torn. I've named Ichnotaxa myself. I'm still, you know, they're not species. They're not genera. Um, they're behaviors. It's the system we have though, and so we're kind of stuck with naming Ichnotaxa. And you know, and I do it. It's had many discussions over beer with Murray, discussing how else could we actually do this. But it is a system that we have, and it won't change. I agree with you about ichthyofamilies. I think that using ichnogenera and ichnospecies works, but I think when we go to ichthyofamilies, we're dipping into something that I don't want to get into. I never ever use ichthyofamilies in my publications. I think that I just have never seen a good reason for it. I think that sometimes naming families is a great reason for someone to write a paper and do this. But the truth is, when we start to get into ichthyofamilies, and we have all of these unrelated traces lumped into an ichthyofamily, what does it give us? What does it tell us? I I don't see any virtue in it. And so I go back to the ICZN. The ICZN allows for the use of ichthyofamilies, but does not require the use of ichthyofamilies. And so I myself, in my writing, never ever use ichthyofamilies. For me, the reason I do ichnology is to try and understand more about the sedimentary geology, about the paleontology, about the paleoecology, about biotic interactions. And I find that when we start using ichthyofamilies, that's just doing ichnology for ichnology. It's not doing ichnology to answer a broader question. Now, there's those who disagree with me, and I'm happy to get into that argument at some point with people. I'd be more happy to do it with a beer in my hand. But for myself, I've never used an ichthyofamily in a paper that I've written myself, and probably never ever will, because I don't see the virtue in using them.
Yes, it's driving us for a different purpose of the technology, that is to identify the behaviors. Thanks so much for answering my questions. Nice, nice presentation.
Thank you, Lucas. Nice to meet you online.
Yes, yes. We are talking with the Ichnos through the Ichnos journal, and I'm glad to meet you online.
Yeah.
Thank you so much.
Any other question? Yes.
Yeah, can I ask a question, please, JP?
Hey, how you doing?
Yeah, all good. How are you? Love the talk. Great to see the glyptodonts. This is something I guess we talked about a little bit with some of the other glyptodonts. But I'm wondering, you mentioned with the gunnel-lignus in the turtles that seems to be you get that in situations where they're stressed. Is that something that you've noticed also with the kind of the other hemispherical marks? Would they be more common in stressed turtle populations? Have you seen any evidence of that? Because that's kind of something we've been thinking about with the glyptodonts as well.
Yeah, that's a great question. So for me, the gunnel-lignus is actually a labor of love. When I was one of Al Holman's grad students, a master's student in 1992, I think it was, Al had me, prepping out turtles. It was a disgusting job, taking, all these winter kill populations and then rendering them down using maceration, so using anaerobic bacteria to get rid of the flesh and then pour it through the screens to keep the bone. And I started to see some interesting traces. And I found a specific turtle that almost 2/3 of its shell had been corroded away by subcutaneous ulcer disease. It was like 2/3 of the turtle was absent, only 1/3 of the shell. How that animal survived, I have no idea. And that got me really interested in that. And that was actually something that Al and I talked about. And as a consequence, I ended up working with turtles from different areas. And one of the things we noted then was that we actually had a better success rate at finding turtles that actually had ulcerative shell disease and subcutaneous ulcer disease by bringing them into aquaria and taking care of them in proper environments rather than treating them with antibiotics or antifungal agents. And so the veterinarians tended to actually prescribe those to people whose pets actually had these diseases, and to people whose ponds were characterized by these diseases. And one of the things we'd noted was the ponds that actually had these problems often had phosphate loading from farmers who fertilize their fields. They had too much fecal material coming in from areas that had been spread with animal waste for fertilizer. So when we had nutrient loading, we were getting more cases of this disease. And by taking the animals and removing from those conditions and bringing them into healthier conditions, they survive better. Right now, where I'm sitting, there's a turtle about 4 meters away from me that was a turtle somebody had as a pet. Same thing. They actually had it in improper housing, too small of a space, improper basking space. And they were treating this initially with antifungal and antibiotic agents, and they weren't helping the turtle. And so the turtle was then dropped off at a pet store, and I took it over as a rescue turtle. I treated it with nothing besides a better situation. And so the tank right now is 2 meters long and 3/4 of a meter wide. She has, April, the turtle, has amazing basking space, and she's healed now. She's fully healed. She still has a scar on her shell, but she's healed because of that. You asked the question about the hemispherical pits. Yeah, I do think that. We've noted that turtles, again, in the Trachemes paper, after we wrote the Trachemes paper, we ended up getting hold of some data from areas in which we actually had wastewater input from one was from a sewage treatment plant, one was from a power generating plant where the water became too warm. And again, in both those areas, compared to the adjacent Trachemes populations, the amount of pitting in the shells was astronomically higher. Now that'll be a paper we write someday. Get off my butt and do it, like many papers. But yeah, so that's actually something that we're seeing in turtles. Stress conditions result in higher shell penetration. Now, the problem we have with the hemispherical pits in the turtles is we can't actually truly limit what we're seeing to an increased susceptibility to parasitism. It might also be an increased susceptibility to infections, to bacterial, fungal, and whatever else. And that's something that we have to try and address as well. And one of the ways we're gonna have to do that, and it probably won't be me, but one of the ways I encourage one of our younger members out there is start making observations. Go out into natural environments and trap turtles, see where we actually see increased amounts of leeches attached to traces, and are we seeing in the same areas increased pitting in the shells? But certainly, a lot of the pits are probably the result of leeches in aquatic turtle populations, but I think that in some cases it may also be other things as well. Certainly, when we talk about that gunnelicness, the third type, when we're seeing those traces, it starts off as on the carapace. It starts off as circular attachments of algae, in a very specific form of algae, Arnoldiella. And then it starts to actually become these linear trends across the turtles. Now, why the algae attaches in lines, I don't know the answer to, but it seems to do that. And we're seeing scarring both in modern and in fossil turtles. Obviously, in the modern, we know it's related to the algae. In the fossils, because we're seeing in this one group of turtles from two different basins, from one genus, Baptomys, we're seeing these linear trends only on the carapace. I think it's got to be an algal agent that's causing the problems. But yeah, like I said, as an observation, and it's one of those ones. In fact, that's why you and I met. I was spending time at the University of California Museum of Paleontology, looking at their turtles to try and expand our knowledge set. We're not going to be able to answer the question on what's happening with the abundance of pits temporally until we look at a lot more turtle specimens. And so at this point, I've only looked at, I don't know, 3,000 or 4,000 turtles. I need to look at another 10,000 before I actually have answers to your questions. Good to see you, Oscar.
Yeah, you too. Thank you.
Yeah.
I'm sure we'll talk about this offline as well.
Sounds good. Yeah.
Okay, any other question? Nope. Well, thank you very much. It was really a great talk. I have learned a lot. And thank you everyone for being here. We are going to meet again in the last week of November for our last talk of this conversation series. So thank you very much and have a great weekend. Thank you, GP.
Thank you, Nelia. Thank you, everyone, for listening.
Thank you, GP.
Good to see you, Renata.
Good to see you again. Bye-bye.
Take care.